Hi, everybody. This is Mr. Yeoman, and today we're going to be taking a look at gene expression. Previously, we had talked about DNA and know the importance of DNA, but we have to be able to get from the concept of DNA to actually expressing that, making that cause our body to work in certain ways. And so sometimes gene expression is also referred to as the central dogma, which is just the process of going from DNA to the way that your body is functioning. And so another way we can think about that is genotype, which is a term that we use a lot in genetics. So we'll talk about that more as we move into genetics. That genotype is really that DNA structure. So, what are the alleles that were given to you by your parents? What does the DNA coding say? And how do we get that into an actual functioning? How does that make our body work? How does that make us appear the way that we appear? And so, since our body really functions off of proteins, DNA codes for those proteins. And so, how do we go from DNA to proteins? Since those two molecules are really quite unrelated, we have to figure out how do they end up having that connection. And so in this first drawing, what I have is a segment of DNA. So let's say that it's a single strand off of a DNA molecule, and I have the end sort of broken here because this would be part of a long continuous strand of DNA. But I have it broken there because we only want to focus on a single gene. When we're taking a look at DNA, a lot of times they're part of chromosomes, and those chromosomes are going to have multiple genes on them. And so we just only want to take a look at one gene because this one gene is going to be coding for one protein in this case. And along with that gene, we have a couple of regions. So we have a promoter region. That promoter region is what is going to call over the RNA polymerase, saying, hey, there's a gene that needs to be coded here. And then at the end, we need to know where this gene ends, and so we have the terminator region. Those will become a little bit more relevant as we talk about transcription in more detail. Okay, so gene expression is broken up into two major steps, and the first one is transcription. And so if you think about what the word transcript means, or if you think about history, scribes would write things. If you think about the transcript of a phone call, it's the entire phone call that is written out as, here's what people said. So transcription is just rewriting. And so what we're doing is we're copying DNA into RNA. But why do we want to do that? Okay, why not just make another copy of DNA? And here's why. The DNA is so important that we don't want it to be damaged, because if it becomes damaged, that is our original copy. And so any changes are going to end up being carried on into any other cells that are made from that DNA. In this case, since we're just trying to make proteins and not make entire copies of the cells, if we make a copy and that copy ends up becoming damaged, then we can just go back to that original DNA and make a new copy. We don't want to take our only copy of DNA, take it out, and then end up having it get damaged along the way and not know what's damaged. We just want to be able to take that copy out, and if that copy gets damaged, not that big of a deal. The second reason that we want to make RNA instead of DNA is that ribosomes, which are the organelles that actually make the proteins, only read RNA. They don't read DNA. And there's a whole history about that, and that we'll take a look at in some later videos and take a look at later in the class. But most biologists think that before DNA, only RNA existed, and that's backed up with a number of pieces of evidence that we'll take a look at in more detail at a later point. But essentially, that's why we need to make RNA, is that we want to protect the DNA, but then second, we want to be able to have the ribosomes read the RNA. They're not able to do that to the DNA. Okay, so during this transcription process, there are some other regions where we have these regulatory proteins. Some genes we want to have running all the time. Okay, those are the housekeeping genes. So a lot of the things that are related to metabolism, making insulin, things like that, things that are pretty regular. But sometimes we don't need to make genes all the time, or maybe we just need to make them a lot in very, very short bursts. And so we have these regulatory proteins that will either tell us stop making that protein. So it pretty much interferes with the RNA polymerase ability to copy that DNA and turn it into RNA. Or you can have activators, which are going to do the exact opposite, saying, no, make more copies of this. Okay, and so depending on the situation, you either want to shut that down, shut down the production of that particular protein, or you want to increase the production of that particular protein. All right, so once we have copied that segment of the gene, and just like with DNA, we're copying it. We have that 5' to 3' section because we can only add new nucleotides to that 3' end, so we can only copy it in that direction. So we have that copy. Well, now we need to actually be able to make the protein, and so that's our second step. That's translation. And so essentially what's happening during translation is that we're going to use that RNA that we copied during transcription in order to provide the directions of what polypeptide chain, what amino acid chain are we going to build. And so now we have this messenger RNA, and messenger RNA is just that RNA after it's been processed and manipulated. We'll talk about that more in a later video. So there's some splicing that sometimes goes on. There's some other processing that occurs. But that messenger RNA is going to go over to the ribosome, and that ribosome has two parts, and that messenger RNA is just going to get sandwiched in between these two parts. And the ribosome is going to read that messenger RNA and say, all right, what's the next amino acid that is going to be added to this chain? It just keeps doing that until it gets to the end of that messenger RNA. So you could end up with hundreds of amino acids just listed one after another. And once it gets all done, then that polypeptide chain is going to be, all strung out in this long line, but then it's going to start to fold. So if you think back to the macromolecules unit and how proteins end up having these different levels, that primary, secondary, tertiary, and quaternary structure, it's going to start folding up and making that protein. And so once that protein is made, then that is going to go out and do whatever job that is. So if that is to provide pigment to your skin, it's making that protein, that melanin. Or maybe it is making a protein to help you with carrying oxygen in your blood, so related to hemoglobin. So you have these different proteins that they're going to be doing different jobs. Now, to clarify, again, I said that messenger RNA sometimes gets processed. In prokaryotes, that's not the case. In prokaryotes, that messenger RNA goes straight to the ribosome to get translated. In eukaryotes, because we have a little bit more complicated relationship with our environment, they have to have these things added to them. So we have these poly-A tails. So they have to go through some additional processing first. But at the end of the day, they're both going to do the same thing. They're going through the same steps. Those steps might be slightly different, but they're essentially doing the same job. All right, well, I hope that helped. And as always, stay curious and seek truth.